all the way from Finland, Herantis and Pekka. Although a Finnish company, quite a strong investor network, institutional anchoring in Sweden, and pretty much you'll be here more than you're in Finland, I hope. Please take it away.
Thank you, Oman. That's correct. So very excited to be here. Very happy that the language is English today. I'm the CEO of Herantis Pharma. My name is Pekka Simula, and som ni vet, om man heter Pekka så talar man dåligt svenska. But as Oman said, I've been frequently in Stockholm recently and continue hopefully to be so as we are currently under our Swedish IPO. So Herantis Pharma, toward clinical breakthroughs. So we are trying to do something that hasn't been done before, facing unmet clinical needs. Our target indications are Parkinson's disease, an incurable progressive brain disease, and then secondary lymphedema. A picture shows more than 1,000 words. So this patient here has breast cancer associated lymphedema, in which lymph, the liquid, starts to accumulate in the arm because breast cancer treatments have damaged the lymphatic system of this patient. So as you can see, it's a very painful, significant impact on the quality of life of the patients. So two significant unmet clinical needs. Just out of curiosity, how many among you know someone with Parkinson's disease or someone who's had breast cancer surgery? Oh my God, that's a big number. This is actually really true. Parkinson's disease impacts about 7 to 10 million people worldwide. And even though there are Parkinson's drugs, they can only treat the motor symptoms of the disease for a certain time, and even their efficacy is lost to disease progression. And it's also a very expensive disease for societies. It's estimated that the societal costs of Parkinson's disease are over $50 billion every year. It's estimated in the United States that if there was a treatment that could stop progression of Parkinson's disease, it would save the society almost $500,000 per patient in the United States. The same is true of lymphedema. It's been estimated that those breast cancer patients who are cured of their breast cancer, but as a consequence they develop secondary lymphedema, they will cost the society between $10,000 and $20,000 every year in the US through the rest of their lives. Not to mention just the human suffering, but also very expensive diseases to the society. And this is exactly what we want to change. So we are aiming to develop disease-modifying treatments. We don't just want to treat the symptoms of the disease. We truly want breakthroughs in the treatment of these diseases, so we could stop disease progression or, in case of lymphedema, even cure the patients. So if we go back to Parkinson's disease, this is probably a gentleman many of you remember. This is Professor Arvid Carlsson, and his work at the University of Lund have been groundbreaking to the current treatments of Parkinson's disease. And starting from the 1950s, this led to Parkinson's treatments with levodopa from the 1960s, and it's still the golden standard of Parkinson's disease 50 years later. It tells you something about how challenging a disease Parkinson's disease is. So it would be about time to get something better for Parkinson's disease. And that's what we are trying with our CDNF. It's a protein that we all have in our brains. It's a natural protein whose role in our brains is to help our neurons stay alive. That's of course very important because we don't get any new neurons since our childhood. And CDNF seems to do this very, very efficiently in healthy brains. But when there's a disease pathology, such as Parkinson's disease, that starts to kill certain neurons, in that case, the dopamine-producing neurons in our brains, then the natural, very small, low CDNF levels are not sufficient. So obviously, it's been a very natural idea since the groundbreaking discovery of CDNF to harness its potential in a disease. And Parkinson's disease would be a very natural target. So this is the most scientific slide I'll show today. Apologies for this. In this picture, the big thing here is a neuron. The small thing here is a glial cell, the immunological cell in our brains. And CDNF works naturally in many different ways that are very relevant also to Parkinson's disease. This here is the endoplasmic reticulum in the neurons, and in neurodegenerative diseases such as Parkinson's disease, there is a tremendous endoplasmic reticulum stress, or ER stress, in these neurons, which eventually drives these cells to the programmed cell death, apoptosis. And CDNF can very potently protect the dopaminergic neurons from this ER stress. It can help them recover from ER stress. But CDNF also directly promotes neuronal survival. It also protects the cells from the toxic alpha-synuclein, which is a common pathology in Parkinson's disease. And it even works through the glial cells and reduces neuroinflammation, which again is a common thing in not just Parkinson's, but many other neurodegenerative diseases. So that's one of the reasons for our excitement, that it works through several mechanisms in a natural way. And in drug development, as you know, you always have to do animal studies before you enter clinical studies. So before the now ongoing clinical studies, this picture is from probably the most important animal study we did. This is done in rhesus monkeys, a primate, a fairly large primate, and a so-called MPTP lesion in the aged rhesus monkeys. So it's a well-established Parkinson's model, and in this model, the monkeys have a very profound Parkinsonism caused by exactly the same thing as causes the human Parkinson disease. So the dopaminergic neurons in the midbrain of these monkeys have lost their function. This is also shown in this picture. This is actually a slice of the brains of the monkey, and the dark staining shows the dopaminergic function in the brains. So as you can see in the picture on the left, almost all of the dark staining, almost all of the dopaminergic function has been lost. As a consequence, these animals are very apathic. They become very immobile because they can't really use their one arm and one leg. So just like Parkinson's disease patients, they have a severe motor disability. And when we treated these monkeys for 3 months with once a month dose of CDNF, we saw a significant neurorestoration. So a lot more dark staining here, a significant improvement in the dopamine function in the brains of these animals. This very nicely correlated with also improvement in the gross motor function, in the fine motor function in the animals compared to placebo group. But also for the first time in this animal model, we saw an improvement in non-motor function. So we even were able to improve the motivation of these monkeys. We were able to reduce the depressive behavior in the monkeys. And these monkeys actually became active again. So since the Parkinsonism, where they just sit apathically in their cage, they started again to react to their caretakers and look for food in their cage and so forth. So this was a very significant finding, of course keeping in mind that humans are not monkeys. Our brains are even more complicated. So as you well know, the predictability of animal studies doesn't tell that it's going to work in humans. But this definitely provided us a very strong basis to move forward to a clinical study. And not just that, it also provided us the basis for a strong EU application. We were actually granted a €6 million Horizon 2020 grant by the European Union for the clinical study. And on top of that, we were able to convince very strong academic sites, Parkinson experts, to participate in this clinical study. As shown, Lund University has since the 1950s been one of the leading Parkinson's research sites in Europe, and now we have Parkinson's. Our clinical study also conducted at the Lund University Hospital, as well as here locally at Karolinska, with 2 very experienced Parkinson neurologists. We're also recruiting patients, of course, in Helsingfors, in our home ground. And the study, even though this is the first time in the world when CDNF is given to humans ever, we were able to do it already in a very exciting setting. So even though it's a first in human study, it's already a phase 1 to 2 study in which we have a randomization placebo-controlled group. So we have fully recruited the study. There are 17 patients, and they have been randomized to receive either placebo or CDNF for 6 months. And then in the first quarter of next year, we expect to have the unblinding of the study and announce the first top-line results from this study. So a very exciting time for the company in this respect. Of course, keeping in mind that it's a first-in-human study, the regulators have required us to do in relatively late-stage Parkinson patients, so they are definitely not the optimal group for a neuroprotective and neurorestorative approach such as CDNF, but that's always what you have to do. You have to start with those most challenging patients. And an important thing also to keep in mind is that after the unblinding, the study will actually continue for another 6 months. So we believe that even after the announcement of the top-line data in the first quarter of next year, more data will continue to accumulate, and we hope that if we see any signal of efficacy, that would then become more robust as time continues. CDNF is a protein, just like many other proteins or many other brain disease drugs. It doesn't penetrate the blood-brain barrier, our protective layer in our brains, if you will, which protects our brains from anything toxic or harmful in our blood circulation. So many drugs don't actually enter the brains from the blood circulation. And that's why we are using an approach which has been used previously in Parkinson's disease. We have a device like this, so every patient, when they enroll the study, have been implanted this medical device, a sophisticated device which is used for administering the drug CDNF directly in the midbrain of the patient, so directly in the right place where it's needed. This is a well-suited approach in Parkinson's disease, and actually one of the common motor treatment in Parkinson's disease today is a deep brain stimulation, which requires basically exactly the same surgical procedure. But then when we'd want to move forward, maybe considering other neurodegenerative diseases such as Alzheimer's or ALS or Huntington's disease, which also are huge unmet clinical needs, and in which theoretically CDNF also should work or could work because the mechanisms are relevant also to those diseases. Then we'd need some approach that could deliver the drug systemically. We need to get it everywhere in the brains. And that's why we are really excited about the X-CDNF, the next generation CDNF. So it has been shown and recently patented that if we take certain parts of CDNF, certain fragments of CDNF, they actually retain the biological activity of the full protein, but they also penetrate the blood-brain barrier. So we already have an in vivo early proof of concept in a rat model of Parkinson's disease. That's just a simple subcutaneous administration of these next generation compounds gives us the same efficacy as the protein when it's injected directly in the brains. And this, of course, is of huge importance and huge interest to also possible partners, possible pharmaceutical companies who might partner with Herantis, because then this could open up the potential for even bigger opportunities than Parkinson's. That's Parkinson's disease. Then our other assets in secondary lymphedema completely independent science from Parkinson's. As discussed, lymphedema is a disease where the liquid starts to accumulate somewhere, and in case of breast cancer-associated lymphedema, it's affecting the arm. And so naturally, if you'd want to cure lymphedema, then you'd need to reconstitute the lymphatic system in that damaged area. And that's exactly what we are aiming with our Lymphactine. This is based, again, on a human natural approach, a human growth factor called VEGF-C. The role of that growth factor in our bodies is to grow new lymphatic vessels. So very obviously, we administer our gene therapy, which helps our cells produce VEGF-C locally in the area of damage. So in the top picture, the green lines are lymphatic vessels. As you can see, there are a couple of areas where there's a cut, so the lymph doesn't flow through as it should. So we administer a single dose of Lymphactine in that area. That results in formation of new lymphatic capillaries, and those will then mature into a functional collecting lymphatic network. So it will again reconstitute the lymphatic system and hopefully make a big difference for the patient. This has been tested in the preclinical stage in a porcine model. The pig is a very relevant model in this case because it's of a similar size than humans, and it has a lymphatic system that is very similar to the humans. So we've been happy to see a very strong preclinical proof of concept. And this has already advanced to a phase 2 clinical study, again in Finland and Sweden. So we first completed a phase 1 clinical study with excellent safety. We also saw some nice signals of possible efficacy in those patients. But since it was not a controlled clinical study, we of course can't make any conclusions on the efficacy out of that study. So now we are in a phase 2 clinical study, and that's being done at 5 Nordic university hospitals. Here in Sweden we have Karolinska and Uppsala University hospitals, and the target is to have the study fully recruited with 40 patients randomized, 20 in the active, 20 in Lymphactine by the end of this year. And then after a 12-month blinded protocol, we will have the readout. So that will be roughly end of next year. So again, one more very exciting thing we're looking forward to next year. And the lymphedema market is growing and very exciting because it's been very much neglected over the years, and now there's a very strong patient advocacy group, Lymphatic Education and Research Network, and a very vocal spokesperson, Hollywood superstar Kathy Bates, who has breast cancer-associated lymphedema, and she's encouraging patients to stop being ashamed of their disease, but to start demanding better treatments. So, to summarize quickly, Lymphactine now in a phase 2 study, CDNF in a phase 1/2 study in Parkinson's disease, both having readouts in the nearest future. Parkinson's will have top-line data out in first quarter next year from a randomized study, and Lymphactine roughly at the end of next year from the randomized phase 2 study. Plus the non-invasive ex-CDNF still at early preclinical stage, but with a very exciting add-on potential to CDNF. And in the interest of time, I think I'll jump directly to quick financials. We have previously been listed in Finland. Our last funding round was completed in March 2019. The cornerstone investor there, as Homan mentioned, came from Sweden. We have Swedbank Robur Medica now as the cornerstone investor of that round. And we are currently in an IPO in Sweden, so now we are coming also to Sweden, and the announced share price of the IPO, we're selling or issuing new shares for the price of 71 kronor per share. Interestingly, just over the past week, our share price has increased in Finland to between 8 and 9 euros. So compared to the current offering price of 6.65, the situation is rather unusual, I would say. We are surprised ourselves that that's what's going on here. But if you're interested, the subscription period will continue through the end of this week. You can make subscriptions at Avanza and Nordnet. So, and of course, I'll be very happy to provide more information. Thank you.